Okay, very good afternoon, my dear friends. Regarding of CPGET 2024, we are conducting this free online coaching for CPGET 2024, especially for the botany students, for the sake of your benefit. Definitely, my students like you, Srinath, Navi Sultana, Ruchita, Nitya, Mehvin, and other students, and also some of the YouTube followers, they are listening and taking notes from the YouTube class also. We hope these classes definitely useful to you, and also definitely you will achieve your goal, and you may get the seat in university campus, different universities. We hope definitely follow the previous classes. I have already analyzed so many papers, previous papers. So CPGET 2020, 21, 22, 23, I have already analyzed and YouTube sent in the YouTube channel. So my YouTube channel is there. So better utilize that YouTube channel, Dr. Padala Tirupati. That is competitive world TSPSC, Dr. Padala Tirupati. That is my channel name. Better utilize this wonderful opportunity. And also, please suggest very nearer to your friends and the aspirants, those who are going to attend CPGET 2024. Definitely it may useful to you. And these videos, links, and the previous papers definitely useful to your friends also. So this is my sincere appeal to you. And also, our resource persons bear their valuable services for the sake of your benefits. So definitely you may achieve your destination. Listen carefully. During the class, if you have any doubt, please clarify your doubts, and also make your own notes, make your own synopsis, make your own points whenever class is going, or you may write running notes also from the during the class hours, and also every class in YouTube link. So that's why you can also visit that YouTube channel also, no doubt at all. But time is very less. Utilize this fruitful time in a proper and in a rightful manner, my dear friends. Our resource person, Dr.A.KAVITA Madam Garu, Assistant Professor, Department of Botany, Food and Nutrition from RBVRR Women's College, Autonomous, Hyderabad. Since Madam has given her valuable services for the past 4 years, that is 2020, 21, 22, 23. And this is the 2024. So many students, they are achieved their goals. Along with Kavita Madam Garu, Dr. A. Rajni Madam, Dr. P. Manjula Madam, Dr. Padmaja Madam, and Dr. D. Nagaraju, Dr. Gogikar Bhagirath, and some other faculty members, they are rendering their services for you. So definitely you can utilize this opportunity and listen carefully and write your own notes. Madam will going to join within 1 minute, and please wait for 1 minute and listen carefully. Okay. And one more important notice: if you have any doubt, even though the previous classes, you may also ask during the class hours. Madam will going to clarify your doubts. Otherwise, even though you can directly contact or communicate, or even though make a call, definitely I will interact with you. Okay. Each topic, Nafis Sultana, Maria Mehvin, Ruchita, Nitya, Naina, is these questions, important questions, may useful for you. Definitely you can note down, or even though you can read it once or twice, whenever you have free time. Okay. definitely within 2 to 3 days, I will send the remaining all the important questions, multiple choice questions related to our CPGET 2024 in previous papers. Questions also will send in the WhatsApp group. Okay. So, Madam has joined just now. So today, Madam is going to explain about the gymnosperms, especially the Netopsida, the class Netopsida, the order Netalis, so the family Netaceae, and the genus Netum. Madam is going to explain about Netum, general characteristic features, morphology, anatomy, reproduction, life cycle. And even though the features related to the angiospermic features we are observed in this Netum, Madam is going to explain. Madam, please share your screen and start your session. Madam. Please welcome.
Okay, sir. Okay, sir. Thank you. Good afternoon, everyone. So today I'm going to explain about the Netum life cycle. Just I'm start sharing my screen. Yeah, I hope the screen is visible. Right. So this is about the Netum plant, and I'm going to explain about the Netum life cycle. So just let us we know about the historical background of this Netum plant. So according to Chamberlain, in the year 1935, he has observed that there are only 3 genera which belongs to the order Netales and the family Netaceae, that is called as Ephedra, Welwitschia, and Netum. And Florin, in 1933, he advocated strongly for splitting Netales further into 3 orders like Ephedrals, Welwitschials, and Netales. And Foster and Gifford in the year 1959, he classified all these 3 genera into a single family under the order Netum. That is, all these 3 genera, that is Ephedra, Welwitschia, and Netum, these all were comes under the common single family called as Netaceae under the order Netales. Whereas Maheshwari and Vasil in the year 1961, he considered Ephedra, Welwitschia, Netum to be the highest evolved plants among the gymnosperms. And Sporny, this is a recent, that is the one which classification what we follow now. So in gymnosperms in the year 1965, he divided the Netales into 3 unigeneric families. That means to say a single order Netales will be there, and under these 3 orders, 3 families, and each family is represented by only a single genera. That is the reason why we call it as unigeneric families. What are those 3 families? It is Netaceae, the one type study what we are going to study, that is Netum, Ephedraceae, the family, and here it is Ephedra, and Welwitschiaceae, the genera Welwitschia. So this is the Spawny classification followed. Next, what are the general characters of this order? Netales. So this has historical background. Yeah, the general characters of this order, Netales. What does it mean? These are the plants. So Netales, it is going to have 3 families. Unigeneric. So what are the common characteristic features of this order? Netales. So they are mostly woody in nature, and some species are trees like Netum, Nimon. The species and the genera, they are the trees, and many are lions and the shrubs, and a few are stumpy turnip-like, that is Welwitschia mirabilis. And the leaves are very simple leaves, elliptical or strap-shaped, or sometimes reduced to scales, and then generally they are opposite or whorled. So this is the leaf structure you can see here. They are the simple leaves, and these are the seeds of the Netum. Okay, the leaves are simple, they are elliptical or strap-shaped, or sometimes they are reduced to minute scales, and they are generally opposite or whorled. And we can, sir, excuse me, sir, somebody are sharing their screen. Ask them to please switch off the video.
Yeah, okay, madam. Okay, I will take care. You can proceed, madam. You may proceed.
Yeah, right. And the leaves are simple leaf. That is, lamina is undissected. It is having only a single leaf. And the shape of the leaf is elliptical in shape and or strap shape, or sometimes, not generally, sometimes they may be reduced to the scale-like. And they are generally, when we talk about the phyllotaxy, they are opposite or whorled. Now we can see the vessels are going to be present in the secondary wood. So this is an advanced character among the gymnosperms. So this presence of the vessels we can't see in the what we call it as pinus. Okay, the primitive characters we can't see. Now the flowers are unisexual. Okay, that is male flower is different from the female flower, and usually they are dioecious and rarely they are monoecious. Now the male flowers, they are surrounded by a perianth, that is a protective leaf-like structure, what we call it as a perianth. And in each female flower, we can see the presence of a single erect orthotropous ovule is going to be present. And inside the ovule, we can see the nucellus. It is going to be surrounded by 2 to 3 envelopes, or we call it as the integuments. But whereas in the pinus, we have seen the ovule is surrounded by only a single integument. That's why we call them as unitegmic ovule. But here, the nucellus or the ovule, it is remain surrounded by 2, that is bitegmic or tritegmic envelope. Next, we can see the pollen tube contains 2 male nuclei, and a unicellular primary suspensor is observed in the embryo. So these are all the common features belonging to the order Netales. Okay, so this is applicable to Netum, it is applicable to Ephedra, and it is applicable even to the Welwitschia. Okay, now 2 cotyledons are going to be present in the embryo, developing to the 2 ovules. I hope the general characters are clear. Now we are moving on to the GNETUM, the type study. So this is a classification. Plantate, Tracheobionta, it belongs to the superdivision called as Spermatophyta, division Netophyta, class Netopsida, order Netales, a single unigenetic family Netaceae, and the genus what we call it as Netum. Now, so what we'll talk about the distribution of this plant. So Netum, it is going to be represented by 40 species, which is confined to the tropical and the humid regions. And according to the scientist Bharadwaj in the year 1957, he says that Netum, in India, there are 5 species of Netum found. And what are those are? First species, we call it as Netum nemon. So how is this plant? This is a shrubby plant. It was found in Assam or Naga Hills region. And Netum contractum. So here the shrub is scandent, like it is more or less tree and more or less shrub-like structure, which was found in Kerala and the Nilgiri Hills. And Netum latifolium, which is a climber. It is found in Andaman and Nicobar Islands. Netum ula. This is a woody climber with swollen nodal branches, which was observed in Kerala, AP, as well as Orissa. And the last species in India, it is Netum montanum. This is also a climber with slender branches and woven at nodes. That is, they are all these branches they are wound at the nodal regions, and these were observed from Assam, Sikkim, and some parts of the Orissa. So these are the 5 species which were observed in the Indian region by the scientist called as Bharadwaj. Now we are moving on to the characteristic features of the Netum plant. So you can look into here. This is a branch of a Netum plant. So you can see here the phyllotaxy of the leaves. They are opposite, and these are the ovules. Strobili. Now, so when you talk about only the Netum characteristic features, these species are sometimes they are climbers, and some are being shrub and the trees. So just now we told you, look at depending upon the species. Only 2 species, they are either shrubs or the trees, but the remaining 3 species, what we found, they were all the climbers. Now, several stems are articulated. That is, articulated means we are going, those are going to have the remains of the branches. So when the older branches, when they shed down from the plant, the remain of the branches will be there on the stem. So that is called as articulated. So branches here, just like Gnetum, that is a pinus. Here also we come across 2 types of the branches. One is called as branches of limited growth, and another one is called as branches of unlimited growth. Principal. Branches of limited growth and the branches of unlimited growth. Now, the climbing species, when we talk about the climbers, the Gnetum, some plants are climbers. They have the branches of only the limited growth, and also the unbranched, we see only the branch, that is only single branch, that's we call it as the unbranched with the foliage leaves. Branching cannot be seen in the climbers. So only single type of branch we can see, that too they are of limited growth, and those are with the foliage leaves. Okay. That is, the leaves are very much expanded. And when we talk about the leaves, so you can see here, the leaves are very much expanded. They are large, oval or subovate shape with the entire leaf margin. So this is the entire margin here. And 9 to 10 in pairs arranged in decussate fashion with a reticulate venation. You can see here, this is a single midvein producing the lateral veins, and the entire margin is a 9 to 10 pairs which are arranged in decussate. So these are about the stem and the branches. Next, moving on to the roots. Okay, we can see a taproot system here in this one. So I'm going for the anatomy of the root here. This is the anatomy of a young root. So here you can see a cuticle. Okay, the epidermis, a single layer of epidermis, which is surrounded by a cuticle. And below the epidermis layer, we can see the cortical region. This is the entire cortical region. And there are layers of, this is completely is the cortex. And this cortex is many layered. Okay, you can see all these cells are cortical cells and they are many layered. And the cells of this cortex region, they are polygonal in shape. You can see here the shape of the cells. They are polygonal in shape and parenchymatous. Okay, they are parenchymatous. That is, they are having very thin walled cell walls, parenchymatous. And here, this parenchymatous cortical cells, they are filled with the starch. So that is the reason why we call it as a starchy cortex in the case of Gnetum root, in the young root. Next, below the cortical region here, you can see a pericycle. Okay, the outer layer of the stela region, we call it as pericycle, which is of 4 to 6 layers. And we can see here, this is the primary xylem, okay, which is very, very visible. And these roots are showing the presence of 2 vascular bundles, that is DR condition. So this is one vascular bundle and this is the another vascular bundle. There are 2 vascular bundles. So we call them as diarch condition, and the protoxylem is facing towards the epidermis. So that is the reason why we call it as exarch condition. Okay, so remember here, Gnetum roots, that is the young root. They are having many layers of pericycle, and the xylem is of diarch condition, and it is exarch, presence of 2 vascular bundles. And in endodermis, below the pericycle, you can see your endodermis layer, and this endodermis are with the Casparian strips, which is an again angiospermic character. Presence of Casparian strips in the endodermis is a angiospermic character, what we observe in this Gnetum. So that is the reason why GNETUM is considered to be as the advanced form of the gymnosperms. The next evolution leads to the angiospermic plant. So this is about the anatomy of the young root. Now, as the roots are growing, they transform into the older roots. So in the older root, this is a transfer section of the older root. So in the transfer section of the older root, the primary xylem is indistinguishable due to the secondary growth. So we can see here the development of the secondary growth, that is the formation of the cambium and all that. So primary xylem is indistinguishable due to the secondary growth here. It consists of tracheids, vessels, and the xylem parenchyma. So vessels here again is an angiospermic character. This was not observed before in the piners or general characters of the gymnosperms. What we say, xylem is only having the parenchyma as well as the tracheids. Vessels were absent. But this is an exceptional case where GNETUM is showing the angiospermic character of possessing the xylem vessels. And the phloem, it is going to consist of only the sieve tubes and the phloem parenchyma. Next comes in the tracheids. If you see the tracheids, brass, we call it as bars of Sanio. This is a cells what we observe in the tracheids and which are absent in the vessels. And these tracheids, they are unicellular, uniseriate, bordered pits are present. That means each tracheid is laid one above the other, forming a single row. And this is called as uniseriate condition with the presence of the pits at the border. We call it as bordered pits. Whereas that is in case of tracheids. When we go for the vessels, multiseriate, vessels are multiseriate and bordered pits are also observed. So this is about the anatomy of the young root and the older root. Now when I go for the anatomy of the young stem and the older stem here, so this is a GNETUM stem, older stem, okay? So first we'll look on to the stem here of the younger one, at the stem when it is in the younger stage. It resembles, when we see here, the section cutting of the young stem of the GNETUM, it resembles a typical dicot stem. So what are the characteristic features of a dicot stem here? We can see the presence of a ring of vascular bundles, okay, and occupying the central pith region. That is, we can see a ring of vascular bundles. And how are these vascular bundles? They are conjoint and collateral with a open vascular bundle. That means we can see a clear cambium in the center of the vascular bundle. Many vascular bundles. So you can see many vascular bundles. They are conjoint and collateral. That is xylem and the phloem. They lie on either side of the cambium. And they are open due to the presence of the cambium in between them and endar condition. That is, the protoxylem is facing towards the pith region. This is called as the endar condition. And all the vascular bundles are arranged in the form of a ring. Now, so when we talk about here, we can see the presence of a single layer of epidermis with cuticle and having the presence of sunken stomata. And below the epidermis, we can see 3 layers of cortical region. The layer which is present just below the epidermis, it is of chlorenchymatous type. That is, it consists of the thick wall chlorenchyma, and that is the parenchyma cells which are going to have the chloroplast in them. So we call it as chlorenchymatous cells. And the second layer of the cortex, that is called as the parenchymatous. So this is the parenchymatous cortical region. And the third layer here, this is a sclerenchymatous cortex. So very clearly we can differentiate here the 3 layers of the cortex. This is chlorenchymatous cortex, parenchymatous cortex, sclerenchymatous cortex. Okay. Now, below that, we can see a single layer of endodermis and pericycle. And just now I explained a ring of vascular bundles, which are conjoint and collateral open and endar condition. And the xylem is comprising of vessels as well as tracheids. And phloem is comprising of about only the sieve cells and the phloem parenchyma. Next comes to the older stem. That is, when the stem is growing, secondary growth will take place. And here, the primary cambium, it is very short-lived. That is a cambium which will form during the secondary growth. It is very short-lived. Sclerotic cells are going to be present in the vascular bundles. So you can see here, a ring of sclerotic cells we can see. Secondary cambium will be developing in the different parts of the cortex in the form of, this is a cortical region, right? Okay, this is complete is a cortex. Okay, so secondary cambium, the cambium will be developed in the several layers of this cortex in the form of ring. So this is the first ring, this is the second ring, and the third ring. So many layers. The secondary cambium is observed in the many layers or in the different forms of the cortex in the form of rings. And the secondary wood is comprising of tracheids and the vessels. And the phloem cells have only the sieve plates, perforated sieve plate that we can see in the section cutting. The medullary, here we can also see the presence of medullary rays. We are consisting of polygonal. These are the medullary rays you can see here. So we call these are the medullary rays which are having the characteristic features of polygonal parenchymatous cells. So here in the anatomy of the stem, the question may be asked like this. The anatomy of gymnospermic stem resembling the dicot stem. Example. So in gymnosperm, which stem is showing the resemblance with the angiospermic dicot stem? So that is the GNETUM. So that points you people have to remember. Now, next moving on to the leaf, anatomy of the leaf. So I told you that leaf is a very simple leaf. Okay, the leaves are simple. It is a large expanded region. So here, just like what we study, like the stem characteristic features resembling with the angiosperm. Here, it resembles a dicot leaf. The leaf of this gymnospermic plant, it resembles a dicot leaf with cuticularized epidermis. That is, epidermis will be covered by a cuticle and having the presence of stomata on both sides except on the veins. Okay, you can see the presence of stomata on the upper epidermal region and also on the lower epidermal region. Next, in the center, this is completely the mesophyll tissue, right? It is differentiated into a single layer of palisade tissue and well-developed spongy tissue. Okay, the palisade tissue is only a single layer here. And the remaining everything is the spongy parenchyma. That is the spongy parenchymatous tissue. So this is again a characteristic feature of dicot leaf. Next, a ring of thick-walled stone cells. They are present just outside the phloem and the latex tubes, which are present in the midrib region. So this is a midrib region. So here in the midrib region, you can see the presence of stone cells just outside the phloem. Okay, so this due to the presence of the stone cells, the midrib of the leaf lamina is appeared to be as a harder one. And here we can't see any transfusion tissue. And one more point here, the vascular bundles are in the form of arc or in the form of a curve shape in the midrib region. So you can see here, these are the vascular bundles which are in the form of the arch, or they are in the form of a curve in the midrib region. And these vascular bundles are collateral, conjoint and collateral type. That is both xylem and phloem. They are present on both and the same vascular bundle. And the xylem comprising of tracheids, vessels and the xylem parenchyma. Phloem is with sieve cells and the phloem parenchyma. So this is about the anatomy. Anatomy of stem, anatomy of the leaves. Right. So that is about the morphology as well as the anatomy of the GNETUM plant. Now we are moving on to the reproduction. So now we are looking up to the reproduction here. So the GNETUM plant, it is a dioecious plant. So what is dioecious condition? What is a dioecious condition, ma? Anybody? Dioecious and monoecious. What is dioecious? That is, male plant will be different from the female plant. Okay. Now, so dioecious, that is cones or the strobili. These are the reproductive structures you can see here. These are the strobili. Okay, this is the entire one is a strobili. And here these are the, okay, so these are the ring of the male flowers you can see here. Right. So the GNETUM, it is a dioecious. The cones or the strobili are the reproductive structures. Now, when we talk about the cones, so here it appearing just like a panicle, the inflorescence type called as panicle. The cones will be in the form of a panicle, what we see in the cereal plants or millet plant. So the cones are just like that, and it is going to have a central cone axis. So in both the one male and the female, you can see the presence of the central cone axis with the two opposite bracts at the base. You can see here, these are the two bracts at the base, and at each of circular bracts, main axis, it forms a cupules. So this we call as a cupule. Okay. Now, so the flower-like structures in the collars, the flower-like structures at the commas, it forms a bracts. Now, when we discuss about a male cone or a male flower, so the flowers are arranged alternatively in a different rings. So these are the flowers which are arranged in the form of a ring. Okay, these are arranged in the definite rings above the each collar. So each point here, it is referred to as the collar on the nodal region. So these are also called as the nodal region. So above the nodal region in each collar, we can see the presence or development of the male flowers. Okay, in a ring-like structure. Next, each flower here, each male flower is going to consist of two coherent bracts which forms a perianth. So just like what is a perianth? It is a protective layer of the flower that we call it as a perianth. Now, above the rings of the male flower, there is a ring of abortive ovule. Okay, there is a ring of abortive ovule. So at maturity, the stalk elongates such that the anther comes out of the perianth sheath. Now, next comes to the female cone. So this is a abortive ovule. These are the male flowers, and this is a abortive ovule. Now, next comes to the female cone and the female flower. A single ring of 4 to 10 ovules are observed above the collar, and only few ovules they directly develops into the mature seeds. Okay, so on a female plant, here also we can see the presence of a panicle-like strobili or the cone, and at each node, above each node, we can see the presence of a collar, and above the collar, we can see the development of 4 to 10 ovules. Above the collar, at each node, you can see 4 to 10 ovules, and among these 10 ovules, only few ovules they develop into mature seeds. Not all the ovules develop into the seeds, only few ovules. And here, when you talk about the structure of the ovule, this ovule internally it consists of a nucellus surrounded by 3 envelopes. And the nucellus is a region which we can see the presence of female gametophyte. And the inner envelope, it will elongate to form a micropylar tube-like structure. And the ovule is considered to be as the orthotypous ovule. Okay, orthotropous ovule. Now, next we move on to the microsporogenesis. That is how a male gametophyte is going to be developed from the spores. Now, so this is the two archisporial cells. You can see here, these are the archisporial cells which are developed in the male flower. These two archisporial cells, they divide to form a multi-layered or multi-celled archisporium. So you can see here the two archisporial cells of the male flower, that is inside the male gamete, that is in the that is inside the male flower. It will undergo division and forms a multi-celled or multi-layered archisporium. Now this archisporium, the cells of this archisporium divides periclinally, forming the so the cells of this, it will divide periclinally and forms the outer layer of the parietal layer or the parietal cells. So you can see here, this is the parietal layer, and the inner layer, we call it as a sporogenous cells. All these are the sporogenous cells. I repeat this once again. Two archisporial cells of the male flower, it will undergo many divisions and forms a multi-layered archisporium. Okay. So these multi-layered archisporium, it will undergo the periclinal division and forms a outer layer of parietal layer and the inner layer of the sporogenous cells. Now the parietal cells here, it is going to form the wall layer of the tapetum and tapetal cells periclinally. Now this sporogenous cells here, whatever we can see here in the center, these sporogenous cells, they undergo division irregularly and forming microspore mother cell. MMC here, it stands for microspore mother cells. That is, the sporogenous tissue, it will undergo irregular division and forms microspore mother cells. And each microspore mother cell, again, each in the sporogenous tissue, each microspore mother cell, it will undergo reduction division and going to form a spore tetrad. So this is a spore tetrad. What is that spore tetrad? Microspore tetrad. 4 spores. They are joined together. So we call them as microspore tetrads are going to be formed. Now, so microspore mother cell, it forms haploid microspores by meiosis, and the wall cells and the tapetal cells of the tapetum, they will get disorganized. Now these are the spore tetrads. Now as soon as the anther, it is going to dehisce along the double row of small cells which extend, all these microspores are released. So this is the anther. So in the male flower, it resembles like a male flower having the male anther. So when the anther is going to be dehisced, all the spore tetrads, the microspore tetrads, they are released into the environment. Okay. So now, this is the development of the male gametophyte we are telling you here. That is, when these microspores, they are released into the environment. So this is a microspore, one of the tetrad here. This is a microspore. So the nucleus of this microspore will divide and forms a 2 nuclei. One nuclei is called as the germinating nuclei and another one is called as the tube nuclei. Now this germinating nuclei again will divide and form the upper basal cell and the lower germinating cell. Okay, this tube nucleus is going to form the pollen tube. Okay. So it depends upon how the cells are going to be formed. So germinating nuclei is going to, we call it as prothalial cell also. The generative cell will form the body cell and the prothalial cell. The generative cell will form the two male nuclei or the two male gametes, which will be projecting into the pollen tube. Now, when I go, that is about the microsporogenesis. That is how two male gametes are formed in the two male nuclei. That is the two male gametic nucleus which are developed in the microspore. Now we go on for the megasporogenesis. Unlike microsporogenesis, here also the female flower, two archisporial cells, they divide periclinally here. These are the two archisporial cells which are dividing periclinally and forming the outer parietal cells and the inner sporogenous cells. Now the parietal cells and the epidermal cells, it forms the periclinally and anticlinally, forming the nucellus. And the sporogenous cells, it undergoes repeated division and form megaspore mother cells. Now this megaspore mother cells and the nucellus you can see here. The megaspore mother cell undergo reductional division and forms a tetrasporic embryo sac. Okay, the meiosis means it undergoes, it forms 4 gametes. Now out of these 4, 3 are going to be degenerative and 1 will remain functional. So that is called as the, 1 second. Hello. Okay. Right. Out of which 1 remains functional and other 3 will remain, they will get degenerative. So finally, the functional embryo sac, which is going to be formed, I have border at micropylar end, tapering at the chalazal end. So this is how the 4 megaspores, tetrasporic development. So 3 are going to degenerate and 1 will remain functional here. And this functional megaspore will develop the, what we call it as a female gametophyte. Now we're going on for the pollination and the fertilization. Pollination here, the pollen is going to be pollinated by anemophily, that is the microspores which are released from the male plant. They will be traveled through the air and they will reach the micropylar tube of the female flower, and thereby the male gametes which are going to reach the egg chamber which is present inside the functional megaspore. Now the fertilization here, the pollen tube will break into the ovule, releasing the male gametes, that is a male nuclei. So one cell will enter into the egg cell and result in the formation of a zygote. Now the another cell will fuse with the another megaspore. Here we are going to form the endosperm. So in GNETUM, cellular endosperm is formed after the fertilization, although cell formation starts after fertilization. So this is again a characteristic feature of angiosperm, development of the cellular endosperm. Now, after the fertilization, a wall formation starts from base and it proceeds upwards such that the cytoplasm divides into many compartments and it is many nuclei which fuse to form a single nucleus. So the upper portion remains free nuclear while the lower portion becomes cellular. So this is how the endosperm is going to be developed. So although we can say here there is no triple fusion, it is a characteristic feature of angiosperms. We can't see the triple fusion here. Only a single fusion, the one male gamete fuses with the cell and forms a zygote, and the remaining all the cells of the megaspore, it will transforms into the endosperm. There is no triple fusion observed here in the GNETUM. Now the embryo development here. So in GNETUM, different species shows variable embryonal development. So according to Maheshwari, the first division of the zygote here in the angiosperm is accompanied by the wall formation, while in gymnosperms, they are free nuclear division. So there is no division of the cells here. So thus GNETUM forms a connecting link between gymnosperms and angiosperms as it shows both free nuclear as well as cell formation. So these are about so at the end of the pushing here, the stem tip with two lateral cotyledons are formed at the tip of the embryo, while the root tip with root cap is formed at the opposite side. Thereafter, the feeder develops between root and shoot, and thus the resultant mature embryo. A mature embryo consists of a stem tip with two cotyledons, a root tip with a root cap respectively. So this is about the embryo development, how we can observe. So this is about the structure of the seed, embryo, the structure of the seed here. The seeds in the GNETUM, they are oval in shape, green to red in color, and it is surrounded by 3 layers enclosing the central endosperm as well as the embryo. So this is a part of the seed when I take the section cutting. Now, so the inner envelope, which is of apparent gametous in origin, middle envelope, it forms a hard, stony and protective in between 3 layers, and the outer one, it is fleshy, parent gametous and the colored. So those are the 3 layers of the seed, and the seed germination, it is epigeal type. That is, hypocotyle will elongate, bringing the cotyledons out of the soil. That is, when we see the germination of the seed, we just like the angiospermic plant. The seeds are coming out. That is, the 2 cotyledons are coming out of the ground surface and they develop into the foliage leaves. First pair of foliage leaves produced by developing the plumule. You can see here the development of the leaves and the plumule development. So this type of seed germination, we call it as the epigeal type of the seed germination. Now we go for the economic importance of the GNETUM here. So this already we discussed in the economic importance, but once again I'm telling, the seeds of the GNETUM pneumon, latifolium and ula, these are considered to be as the edible one, which can be eaten after boiling or roasting. Or sometimes people will be eating these seeds by frying in the oil. Next, the young leaves and the inflorescence, that is the younger leaves, they are edible in the form of vegetables. And whereas the fiber of the GNETUM bark, which is high tensile strength, it is used for making the fish nets and the ropes. And the paper is also being produced from the pulp of the bark of GNETUM pneumon. Whereas a fixed oil, which is used for illumination and also for effective in rheumatism disease, which is obtained from the kernels. Kernels means from the inflorescence of GNETUM ula. So it is widely used in Kerala and the Assam, in the Ayurvedic medicine of Kerala. Next, GNETUM montanum, it possesses pesticidal properties. So this is the economic importance of the GNETUM. So you can look into this. This is a diagrammatic representation of the GNETUM plant. So here, this is a GNETUM, a female shoot. Next comes to a microspore here. Microsporangia, microsporophyll producing the microspores. And here the female shoot going to consisting of the megasporangium. The microspore mother cells, they form the microspores. And the megasporangium, one will be the functional megaspore acting like the ovule. So microspores, when they reach the male gametophyte, they turn in, they will be falling on the micropyle, and then a pollen tube is going to be formed carrying the male gamete, and this pollen tube enters into the ovule where we can see the presence of the egg cell. So fertilization will occur here, resulting in the zygote. The zygote will transform into the embryo. So this is the embryonal development here. You can see the embryo along with the endosperm, and this is an oval shaped seed. So this is the germination of the seed. We can see developing the roots as well as the plumule and the cotyledons. And after the germination, the plant will develop into an entire plant. So this is about the life cycle of the GNETUM. And the most importance here, these are the resemblances, how the NETUM is considered to be as an intermediate between gymnosperm and the angiosperm. So this is the resemblances here. The first resemblance is leaves are showing reticulate venation. Xylem is showing the presence of xylem vessels. A tetrasporic development of the female gametophyte. As I told you, a megaspore mother cell will undergo meiotic division and form 4 megaspores. Out of that, 3 are going to be degenerated and 1 functional megaspore will transform into female gametophyte. And here there is no archegonia development. Directly, we are calling that female megaspore or develops into female gametophyte, where we can transform it into a ovule. So there is no archegonia. So, and the dicot nature of the embryo. Embryo is developing into 2 cotyledons. Okay. So that is a dicot nature. This is about resemblances, and when compared to the wood, the presence of wood are with tracheids, presence of anemophilous pod of pollination, naked ovules, absence of ovary. So there is no ovary formation, thus fruit is absent. Development of, so this is the resemblance of the gymnosperm, NETUM with the gymnosperms, okay? How NETUM is showing the gymnospermic characters. And this is the previous slide, how the NETUM is showing the resemblance with the angiosperms. Okay, so there is no, here we can see naked ovule. The wood is with tracheids formation. Ovary is absent. Directly fruits are going to absent. Ovules are directly attached to the stem. Development of epithelial cells. Presence of cleavage polyembryony. That is about the resemblances of the NETUM with angiosperms as well as with the gymnosperm. So these are some images of different species of the NETUM. This is NETUM ula. This is NETUM montana. These are all other strobili, what you can see. So this is NETUM nimon. So you can see the leaves here resembling with the angiospermic leaf. This is NETUM latifolium. So just stop sharing here. Yes. So this is about the type study of NETUM and not morphological characteristic features, anatomy, reproduction, development, fertilization, pollination, life cycle, and then economic importance and resemblances. I hope this is clear.
Yes, Nitya, Mehvin, Vijay, Ruchita, Nafis. Any doubts? Nayana? Okay, madam, thank you very much. So
Thank you,
tomorrow
sir.
we will meet next class. Thank you very much.
So what will be the next class? I hope all the gymnosperms I have completed.
I will tell you. In the
Okay, sir. Fine.
Right.
Right.
Thank you.